Dr. Ruffell received his Ph.D. in immunology at the University of British Columbia in 2008, and then completed his postdoctoral studies investigating the impact of macrophages on response to therapeutic interventions at the University of California, San Francisco, and Oregon Health and Science University. Since 2015, the Ruffell Lab has been focused on regulation and roles of dendritic cells in controlling the development of breast cancer and in dictating the efficacy of chemotherapy and targeted therapeutics. And the Ruffell Lab is focused on discovering how macrophages and dendritic cells regulate therapeutic efficacy in hormone-driven malignancies, with the ultimate goal of developing approaches that enhance anti-tumor immunity. So we are delighted to have you as one of our first speakers in our first year. So I will turn this over to you, Brian. Thank you.
Right. Thank you, Jen, for the intro, and thank you for the invitation to speak today. So I'm going to focus, I think I'm the first one that's going to focus on dendritic cells from the series, although everyone here is going to see a lot more dendritic cells in the coming few months with Dr. Murad and Dr. Pate speaking. So we'll get this be kind of a really brief intro into the field. Sorry, I'm just trying to get the slides to move. There we go. So I'd like to start off with an intro, just the classic cancer immunity cycle, to really highlight what we think is missing from a lot of research, or at least is only starting to gain traction. And that is that the entire cycle is really dependent upon dendritic cells. So the process of some kind of cell death to induce dendritic cell maturation or activation, the ability of these cells to migrate into the lymph nodes, and then initiate the entire cascade of CD8 or CD4 T cell activation and movement into the tumors, and then hopefully for cytotoxic killing. But while we have, there's a lot of focus on getting these T cells into tumors and to recognize and avoid mechanisms of immune evasion, comparatively less focus on understanding how dendritic cells are involved in this process. And in particular, our lab is really interested in trying to understand what signals drive this maturation process. We're also starting to get interested in understanding how migration of DCs will really impact how therapeutic, how patients respond to therapy. In particular, in light of some recent studies from the Tyler Jack's lab, as well as Nick Joshi's lab, showing that a resident population of stem-like cells in the lymph nodes for CD8 T cells is required to sustain response to therapy, and that dendritic cells may have an important role in this process. What I'm going to talk about today is primarily a kind of a less appreciated role in terms of this cancer immunity cycle, and that is the idea that these DCs don't all migrate out into lymph nodes, but actually most of them maintain their residence in the tumor itself. And there they may have really critical roles in regulating how tumors respond to therapy. And this has been shown now for mediating CD8 T cell responses to chemotherapy and to checkpoint blockade, and really a series of bodies of work, I think, starting in the last about 10 years or so. Starting with work from the Crummell lab, for example, moving into work from the Gajewski group, and as well as more newer work by Pate, Luster, and Mempel. Really defining how the ability of these cells to produce key cytokines and chemokines produces a nice synergistic feedback loop that allows CD8 T cells to maintain and to then kill and result in curative therapy, at least in animal models. And now for the postdocs here, I just want to point out that if you look at the first authors here, we've Spranger and Garris and Picolato. All these are now independent investigators, so this is a really good field if you're interested. There's a very high chance of success for a nice paper and then a nice PI position at the end of that. So moving on. So really centering dendritic cells as a primary mediator of T cell mediated immunity. We think that that will really help understand how these cells are regulating response to therapy. So before I move on, I really need to introduce that most of the talk is going to be focused on this type 1 dendritic cell. So this is the cell type that's been shown to be responsible for mediating CD8 T cell immunity activation. We'll focus less on the type 2 dendritic cells, which are usually associated with mediating CD4 T cell activation. Although there are caveats to this in the recent literature. So these type 1 dendritic cells really express this defined program that was originally identified by Ken Murphy's group, including expression of BATF3 and IRF8 that defines this lineage quite well. They also express the XCR1 chemokine receptor. They express receptors that mediate uptake of dying cells, including CLEC9A. And in humans, they can be usually identified through CD141 or in mouse, CD103 or CD8. But in addition to these kind of classic factors, one of the most important things that these cells do is they express genes associated with involved in antigen processing and presentation. They express genes associated with cytokines that drive a CD8 T cell and Th1 response. And as I mentioned earlier, they express certain chemokines that appear to be required for mediating CD8 T cell function within the tumor microenvironment. So we were really curious as to whether we could target these dendritic cells by identifying therapeutic potential therapies focused on suppressive pathways, assuming that if these cells were functional, well, then we wouldn't have tumors. So if we have a rapidly growing tumor, there must be some mechanism for the tumor to evade being recognized by dendritic cells and initiating T cell mediated immunity. So for the most of the talk, we'll utilize the classic MMTD-PYMT model generated back in 1992 now. So this model progresses to disease in the first 100 days of the mouse's life, and then to late stage metastasis. Interesting, when we evaluate therapeutic responses in this model system, we find that these mice are very resistant to PD-1 therapy. So if you add a simple anti-PD-1 antibody, shown in blue here, you see no response. In contrast, they do respond relatively well to chemotherapies, including standard of care paclitaxel for breast cancer. So we thought this was a good model. I mean, it was available, but also a really good model to understand how immune evasion was occurring. So we then profiled, and this was work I did as a late stage postdoc in Lisa's lab, showing the expression pattern of different inhibitory molecules on various immune subsets within these tumors. And what readily became apparent was that these type 1 CD103 positive dendritic cells expressed a unique panel of suppressive pathway markers, including TIM3, TIM4, and PTLA. And in particular, TIM3 caught our interest as a more selective in terms of higher upregulation by this population. And then just showing here, when we look at the different immune cells, we see highest expression by this type 1 dendritic cell, but by no means unique expression. So TIM3 was expressed by type 2 DCs, as well as by macrophages, and then a lesser degree to monocytes and other myeloid cells. So it's a broadly expressed suppressive molecule with highest expression by this type 1 dendritic cell that we were interested in. So what is TIM3? So TIM3 is, as I mentioned, a surface receptor. It's been associated primarily as an exhaustion marker and a mediator of exhaustion in immune cells, primarily CD4 as well as CD8 T cells. It was originally identified as a molecule that was upregulated on interferon-stimulated CD4 T cells by the Kutcher lab. And then over the years, it's worked out that this TIM3 receptor, the first ligand for this identified was galectin-9, although this is obviously not a specific receptor ligand pair, given that galectin-9 recognizes carbohydrate moieties on receptors. The next ligand identified was phosphatidylserine, although this interaction is incredibly weak, and really it's TIM4, so the family member TIM4 that has roughly tenfold higher affinity for phosphatidylserine. And then more recently, the same lab group, the Kutcher lab, identified CCAM1 as a ligand for TIM3. And in this case, this is now a ligand on either tumor cells or other APCs or other immune cells that can then bind to and initiate a signaling cascade within T cells that promotes their exhaustion or their cell death, or just inhibits T cell receptor signaling. So based upon this, the role for T cell exhaustion, there's been a large panel of TIM3 antibodies that are in clinical development. The two that are furthest along are this one for Novartis, which is now primarily moved into being focused into hematological malignancies, I believe primarily AML. And that's actually based upon TIM3 is also expressed on AML cells, And this, for some reason, appears to mediate nice responses in patients, although the mechanism of that is still either unknown or at least unpublished. The other antibody furthest along is the one originally from Tesaro, now owned by GSK, and that's progressing along in primarily lung cancer patients in combination with PD-1, mostly in PD-1 refractory patients, but there's also trials in, for example, melanoma and in the liver. And these other antibodies are much earlier stage in development, and we're not clear whether these will be functionally relevant or how far these will progress in the clinic at this time. But this idea of targeting T cells with another exhaustion marker other than PD-1 is obviously a very popular topic with the recent success of the anti-LAG3 antibodies in melanoma. It obviously can work very well with minimal toxicity when you're targeting other inhibitory pathways, with the exception of CTLA-4. So this targeting, this biology, at least in solid tumors, was really based upon the very clear expression of TIM3 on exhausted T cells. So this is a paper, one of the earlier papers identifying TIM3 and showing that it could be a therapeutic target in animal models. And even now, 10 years later, showing the same MC38 tumors. You see that in both CD8 and CD4 T cells, the cells that express high levels of PD-1 also express high levels of TIM3, really marking this as an exhaustion marker for these cells, and explaining why when you add TIM3 to PD-1 therapy in these marine models, you get more enhanced tumor regression. However, when we looked at our mammary carcinoma model, we saw this was not the case. We had no problem identifying PD-1 expressing CD8 T cells, but really there was no evidence, if this was an exhaustion marker, no evidence of immune exhaustion in these cells. Suggesting that if we were doing a TIM3 therapy, we really are not acting through these populations. And most importantly, this pattern of expression was very consistent between mouse and human disease. So we did IHC for TIM3 in breast cancer samples, and you really see this stromal staining that can be primarily identified in dendritic cell or macrophage type cells by their morphology. Co-staining for markers, including CD163 for marking dendritic, for marking TAMs, or for LAMP3 to mark dendritic cells. You see nice co-staining of this expression. And finally, we did the same thing by flow cytometry, showing that in the context of breast tumors, as well as just in peripheral blood cells of healthy donors, we see that TIM3 is nicely expressed by dendritic cells, monocytes, and macrophages. And importantly, in these patients, we saw very, very minimal expression of TIM3 on either CD4 or CD8 T cells. And if anything, we found lower expression shown in red here on the tumors T cells than we did in the peripheral blood of healthy donors. So does TIM3 actually have is it a functional target for therapy? Well, we added this to TIM3 blocking antibody in combination with either alone or in combination with paclitaxel chemotherapy. We also threw in an anti-TIM4 antibody to hedge our bets in case we saw some combined efficacy in these early experiments. We found that antibody treatment alone had no effect in this PYMT in these transgenic mice. But when we combined this anti-TIM3 and in this case TIM4 combination therapy with paclitaxel, we were able to really block tumor growth for the duration of the therapeutic treatment. We then found that this was the same as the case in this kind of luminal B type PYMT model as it was in a triple negative model. It's the transgenic C3.1 tag model of breast carcinoma. And again, when we combine paclitaxel chemotherapy shown here in red with this combo antibody treatment, we had a nice suppression of tumor growth. We then broke this apart, treating either these tumors with either TIM3 or TIM4. And we could see that just TIM3 alone was very productive at this effect. TIM4 blockade had no phenotype in this experimental system. So we were hoping to target dendritic cells. Do we actually need dendritic cells to have a therapeutic response in this case? We acquired BADF3 deficient mice from the Murphy lab, backcrossed them on the FEBN, and then evaluated whether we could see therapeutic responses in the transgenic mice. As expected, given the important roles of these cells in driving T-cell mediated immunity, we found that the mice were lacking BADF3 dendritic cells, showed no efficacy in terms of the combination therapy. Then we extended these out to a Black 6 model, an implantable model, using IRF8 flox mice combined with a CD11C Cree. These also result in a lack of dendritic cells. And we found that, as expected, that these tumors, the controls respond fine, but when you lose dendritic cells, you no longer have a therapeutic response. Importantly, we didn't want to just ablate T cell function and then evaluate response to therapy, because if you don't have a dendritic cells there prior to tumor implantation, you obviously don't have a CD8 T cell response to mediate any kind of therapeutic efficacy. So we then turned to an inducible depletion model using the transcription factor ZBTB46 DTR. We could then allow tumors to develop natural immunity and then deplete dendritic cells just prior to therapy. And importantly, in this context, again, wild-type control mice without treated with diphtheria toxin respond as we expected. But if we deplete dendritic cells just prior to initiating therapy, we now lose the efficacy we're seeing, suggesting we were not just eliminating this T cell response in these mice. So what are we doing to these cells to mediate this type of, this improved reduced tumor growth? We already knew that this effect we were seeing was entirely CD8 T cell dependent, using just a simple CD8 T cell depletion experiment or blocking, neutralizing interferon gamma. We can see here that the combination efficacy with TIM3 was completely ablated when we had no CD8 T cells or no interferon gamma present. We therefore thought to look whether these dendritic cells were perhaps presenting more antigen to these CD8 T cells. But we found when we evaluated using a Z-screen uptake model originally designed by Max Krummel, we found really no change in the fluorescence intensity of antigen uptake by these cells. And consistent with several other labs, we found really type 1 DCs were very, very bad at taking up tumor antigen, although obviously this is sufficient to initiate anti-tumor immunity. And then next, we thought to evaluate lymph node trafficking. So again, using the Z-screen mice, we could then track the movement of these cells into the draining lymph nodes, and then evaluate whether we were altering lymph node trafficking in these mice. And then somewhat surprisingly, we found that not only were we not increasing trafficking, but shown here in the blue here, we actually had less Z-screen positive events occurring in the draining lymph nodes during TIM3 treatment. So we were potentially limiting the response to therapy we were seeing in these mice through this pathway. And we're now seeking to evaluate whether this has maybe this relates to the durability of response we're seeing in these mice. So for example, if we actually induce lymph node migration in combination with TIM3 or PD-1, maybe we can get more durable responses in these systems. So what instead was happening was we went through and sorted out dendritic cells, evaluated their gene expression profile back in the day by nanostring, pre-RNA-seq or single-cell RNA-seq days. And we found that out of 500 genes expressed by these cells that we were evaluating, really only 3 or 4 were changed significantly. And these included CXCL9, CXCL10, and CXCL11. So CXCL9, just focusing on this, we saw that both in the CD11C, CD11B positive DCs, as well as in the CD103 positive DCs, we saw roughly a 2- to 3-fold increase in expression of CXCL9, whereas these cells express much less CXCL10 and CXCL11, but really suggesting that we were dealing with an interferon-induced effect in this experimental system that was broadly activating these interferon responsive genes. So we found the same thing was true in vitro. So we isolated splenic DCs, which express very high levels of TIM3, shown here for both the CD8 and CD11B population. When we isolated these cells out and treated them with TIM3 blocking antibody, nothing really happened. However, we developed a system where we would actually kill tumor cells either through just using heat shock, so high heat, or through irradiation and then allowing the cells to die over the course of 24 hours. If we took that supernatant and then added it to our mixture of cells with or without the antibody, we saw that the combination of dying tumor cells and TIM3 blockade led to a nice upregulation in these splenic DCs. And this was true for both the CD8 alpha and to a lesser extent for the type 2 DCs. So based upon this, we went to evaluate whether this pathway of chemokine expression was functionally involved in our response to therapy. So we went back to our model system. We added either TIM3 with paclitaxel chemotherapy, or we employed an inhibitor of CXCR3, shown here for AMG 487. And in this case, that inhibitor dosed, unfortunately, twice a day, to the chagrin of my staff, was able to prevent therapeutic efficacy. We then went on to acquire CXCL9 deficient mice to generate a mixed bone marrow chimera. So this allowed us to, again, have the experimental system develop naturally. We could then deplete either half the dendritic cell population and leaving only wild-type dendritic cells, or deplete half the dendritic cells expressing XCR1 DTR and leave the CXCL9 deficient cells. And when we did this, again, we saw loss of therapeutic efficacy, suggesting that dendritic cells, in particular type 1 DC expression, just of this one chemokine, was required for therapeutic efficacy, really implicating this increased expression of CXCL9 as a driving T-cell dependent immunity in our experimental system. So how does this occur? Well, CXCL9 is a chemokine, so we would assume we're dealing with increased T-cell recruitment. We first looked at both the CD8 T-cell effector function. We found increased interferon gamma expression in the combination therapy, although it was only minor, roughly 30 to 40% increase across our different experiments. This upregulation was lost when we did a CXCR3 blocking antibody or a CXCR3 inhibitor. T cells within these tumors express CXCR3, although this was not changed by therapy. However, when we actually quantified the number of CD8 T cells, whether by flow cytometry, which is not shown here, or by just doing IF and whole tumor scans to quantify the absolute density, we saw no change in immune cell infiltration overall, but in particular, no change in CD8 T cells. So we then hypothesized that rather than mediating actual T cell recruitment, perhaps we were dealing with a more complex situation where the dendritic cells were regulating the organization of the immune structure within the tumor. And this was based upon the fact that dendritic cells are not a very prevalent population. They're not the only population expressing these chemokines, which really suggested we're not dealing with mass influx, but rather local regulation of the CD8 T cell and the CD8 T cell and dendritic cell response. So to address this, my unfortunate first graduate student was tasked with developing a system to measure this. So she, after multiple rounds of optimizing staining for looking at dendritic cells as well as for CD8 T cells, and then finally to doing whole slide scanning with a really old microscope, to then generate these two-dimensional maps of the location of each CD8 T cell and type 1 DC within each tumor. These were then mapped in a collaboration with the IMO department here at Moffit, in particular, Dr. Kasia Rezniak, plotted in MATLAB, and we could then analyze the spatial distribution of these cells to determine whether type 1 DCs and CD8 T cells were now in greater proximity in terms of when we had combination therapy. And indeed, when we looked at mice that had been treated just with paclitaxel alone, we could look at the distance between each dendritic cell and the nearest DC, and showing the distribution shown here in red. If we compare that to the distribution we saw when we added TIM3, you see this nice shift towards the left, indicating that on average, CD8 T cells are now in much greater proximity to their nearest dendritic cell in these treated tumors. Importantly, this was not seen when we did the same experiment in CXCL9 deficient animals. And if anything, this curve slightly shifted to the right, suggesting that, again, it's the CXCL9 driven reorganization of CD8 T cells and DCs that is driving this immune interaction after treating with anti-TIM3. So we further evaluated, well, what's happening here? If we're having CD8 T cells and DCs in greater proximity, why is that important? What's driving this T cell function? So the first thing we evaluated was perhaps we're dealing with higher levels of antigen presentation or co-stimulation. We evaluated the number of cells, or the percentage of cells that were within essentially physical distance of each DC. We found there was a slight shift in terms of, after treated with TIM3, more CD8 T cells were in proximity to their nearest dendritic cell, but this change was very small. And importantly, when we knocked out class MHC1 expression, so class 1 expression from these DCs using this mixed bone marrow chimeric approach, we found that these mice responded quite normally to therapy, suggesting that although antigen presentation is required for the initiation of this immunity, it's not required just for this local therapeutic change that we were seeing in the tumor. In contrast, when we looked about 100 microns out, which is roughly the diffusion limit for certain cytokines, depending on how often they're taken up by other cells, we saw a fairly nice shift towards increased number of cells within this distance. So from 40 to roughly 60% of the cells were now within this 100 micron range of an individual type 1 DC, leading us to think that maybe we're dealing with a cytokine-based system of activation. And indeed, when we did the same mixed bone marrow chimera but used IL-12 deficient mice, we found that now we no longer had a therapeutic response, suggesting perhaps that IL-12 production was leading to improved immunity when these cells were able to come into contact. And that's just graphically what I've shown here. We think that in normal tumors, even though dendritic cells express moderate to low levels of IL-12, they really have trouble finding their nearest CD8 T cell. And so these cells are not exposed to sufficient levels of interleukin-12 to drive their effector function. And this importance of IL-12 has been shown in multiple contexts. So for example, the Bate lab showed that IL-12 expression is necessary for a response to PD-1 therapy in the MC38 mice. And that represents a feedback loop between interferon gamma expression and IL-12 for these cells. And we've previously shown back in 2014 now that if we enhance IL-12 expression, we can also promote anti-tumor immunity, in this case by blocking suppression of DCs through their interleukin-10 receptor. But as an alternative we have here, if we just simply increase CXCL9 expression, we're able to locally recruit CXCR3 positive T cells into the space of these DCs, essentially increasing their exposure to interleukin-12 and allowing them to maintain their effector function in terms of increasing interferon gamma expression. And so this is kind of the first part of my talk. I'm now going to move into the second part where we started to dissect out, now that we know what DCs were doing in the tumor, what's TIM3 actually doing to regulate dendritic cell function? So how does TIM3 act as a checkpoint molecule in dendritic cells? So we returned back to our in vitro system where he developed to look at splenic DCs so that we could easily dissect out some of the molecular mechanisms here. And just to refresh your memory, when we add dying cells, dying supernatant from irradiated or heat shock killed cells, if we do TIM3 blockade, we can enhance CXCL9 expression in these splenic DCs, and just in the short 4 to 6 hour duration. We use this system to then look at gene expression in these cells at a 2 hour time point to ask what was changing and what might be driving this phenotype. What we found was after sorting out the CD8 alpha type 1 DCs and looking at the combination of heat shock, supernatant, as well as TIM3, we found as expected these cells expressed higher levels of CXCL9 as well as CXCL10 and CXCL11, consistent with our data from tumors. And the other thing we obviously found was expression of type 1 interferons in terms of interferon beta 1, alpha 1, alpha 2, as well as these other genes which are direct downstream targets of the type 1 interferon pathway. So interferon responsive genes here and shown here. Oh, sorry. So based upon this, we had an easy assumption that we were dealing with, we have interferon responsive genes, we have increased interferon expression. So in some way, TIM3 is blocking type 1 interferon expression in these cells. And just to, as we would expect, if we just add interferon to these dendritic cells, they light up quite easily with expressing CXCL9. So that's just shown here for the CD8 alpha DCs. And we looked at interferon alpha, beta, interferon gamma. They all turn on very nicely, although there is a definite preference for interferon gamma, beta, and to a lesser degree interferon alpha for the ability to induce CXCL9 expression by these splenic DCs. And importantly, when we added an interferon receptor blocking antibody shown here on the bottom, if we did that in the context of this in vitro system, we really lost this upregulated expression that we were seeing. So completely no signature, no CXCL9 expression was occurring when we blocked this type 1 pathway. As we would expect based upon this experimental system where we have primarily dendritic cells and no CD8 or CD4 T cells to provide the interferon gamma stimulation. So roughly at this time, we started going through the literature to understand, well, what would TIM3 may be regulating to activate type 1 interferons and CXCL9? And we came across this nice review by the Gajewski group that came out about a year after they had defined a role for the STING pathway in mediating spontaneous and irradiation-induced anti-tumor immunity. And we were really struck by an obvious parallel. What we were seeing was we had type 1 interferon expression driving CXCL9 and 10 in tumors, suggesting that this could really explain the biology we were seeing in vitro and in vivo. So we did a simple experiment where we added to our system either a DNase or RNase to remove these from our experimental system. We found that adding DNase very easily removed the response we were seeing in terms of CXCL9, whereas adding RNase had no significant effect, consistent with the role for the CGAS STING pathway, which is a intracellular recognition of double-stranded DNA. We then acquired the relevant knockout mice to ask whether we could lose the efficacy or lose induction of response in vivo. So again, looking at these splenic dendritic cells, if we have either knockout of STING or we knock out CGAS, we see completely no responses in these splenic DCs. And more importantly, when we use these same mice as bone marrow donors to generate a large cohort of animals, we could see that if we lose, if we have wild type mice, we get a response to combination of TIM3 and PTX. If we lose either STING or we lose CGAS in the host mice, we no longer see any therapeutic efficacy of TIM3 and paclitaxel chemotherapy. Now, as before, we wanted to evaluate whether we were dealing with spontaneous requirement for these molecules and immunity, or we were requiring only this activation during response to TIM3. So again, taking advantage of our mixed BMT approach, where we mixed either XCR1 DTR with 50% bone marrow donors from STING deficient mice. And here, if we deplete either wild type and leave only wild type DCs, we see a nice response to therapy. If we leave these XCR1 DTR cells alone and these express STING, we can let them respond to TIM3 blockade. But if we deplete out these type 1 DCs and only allow basically the presence of STING deficient type 1 DCs, we no longer see therapeutic efficacy, indicating that STING expression by type 1 DCs is an absolute requirement for the efficacy of TIM3 blockade. In contrast, when we used knockout mice for MyD88 or TRIF or MAVs, all of these systems responded quite normally in vivo. And when we did the same system in vitro, we again saw the same response. So when we stimulate these dendritic cells, they all light up with CXCL9 quite readily, suggesting we were not dealing with signaling from classical TLR pathway or MAVs, which is a receptor for RNA that can be found, cytoplasmic inhibitor of RNA. So we've nicely shown that CGAS and STING pathway in host cells, and particularly type 1 DCs, is required for the efficacy of TIM3 blockade. So how is this actually occurring? So going back to this nice review from Glenn Barber, who was one of the early individuals defining this CGAS and STING pathway, primarily in the context of viral model systems, but also later showing really key roles for the system in the development of autoimmunity. We can see there are really three parts where TIM3 could potentially feasibly regulate activation of DCs. The first is that perhaps when we're dealing with the release of DNA in the microenvironment, I think we all know that we add DNA when we digest tumor cells, because we have a lot of free DNA in the presence of these tumors, particularly after they die. So perhaps TIM3 was regulating the ability of DNA to enter cells, although how this occurs remains a bit of a mystery. Alternatively, it's been shown by several groups now that the intermediate between CGAS and STING, so CGAS produces cyclic cGAMP in response to the presence of double-stranded DNA. The cGAMP is then responsible for activating STING, which involves the movement into the ER, activation of IRF3 and NF-kappa-B, which then leads to type 1 interferon signatures. These then feed back into a type 1 interferon response and induce interferon-stimulated genes, in this case, in our case being, for example, CXCL9, CXCL10. And so it's possible that TIM3, through some unidentified mechanism, perhaps through signaling, could be interfering with the ability of STING to activate the cells. So to evaluate whether we're dealing with signaling, we added either interferon beta or cGAMP to dendritic cells. And then we added TIM3 blocking antibodies to see whether we see any change in CXCL9. But as shown here, both interferon beta and the cGAMP activated DCs and this is unchanged by TIM3 blockade. We also then looked at, in more detail, at whether we're dealing with cGAMP uptake. So it's been shown that tumor cell production of cGAMP can actually, this can then enter into myeloid cells, in particular macrophages, through, in mice, the P2X7R receptor. And then in humans, there's a certain SLC channel that can allow transfer of cGAMP into cells. But importantly, this receptor is required for immune cells to respond to cGAMP. And we found that while this receptor, this channel is expressed by macrophages, monocytes, it's really not expressed at high levels by DCs. So we think this explains why these, at least the murine cells, respond quite poorly to cGAMP stimulation. We also knocked out STING and cGAS from our tumor cells and then generated the tumor cell debris to then eliminate the presence of cGAMP in this experimental system. And in both cases, we found quite equal levels of CXCL9 expression, suggesting we were not dealing with cGAMP production or cGAMP uptake regulation. So that led us to our last possibility, in that we were dealing with changes in the ability of these DCs to take up DNA. And as I mentioned, the Gajewski lab has done some really nice work back in 2014 now, showing that dendritic cells, or at least CD11C positive cells, can take up DNA both in vitro and in vivo. Although in vitro, this often involved a lipofectamine transfection reagent to get DNA inside the cells. At least in vivo, if they EDU labeled the cells, they injected these into mice, they could then isolate them and use image stream to track this, in this case showing EDU here in yellow, the presence of this tumor-derived DNA in these cells. And that's just quantified here at the same thing, showing we could use this, maybe use this experimental system to see if whether DNA uptake by these cells was being altered. So looking at ImaStream, we started with doing that with the splenic cells. Again, being inspired by the lab from, by the Gajewski lab work. We either left cells, either DC type 1 or DC type 2, being marked by expression of CD8 alpha shown here. We left them either untreated, treated with heat shock and the IgG antibody, or treated with a TIM3 blocking antibody, and then tracked DNA uptake by these cells. We could see in heat shock alone, we saw very minimal levels of uptake. So just occasionally the cells would have some small dots of EDU present. Whereas now when we did TIM3 blockade, depending on the experiment, see anywhere from 30 to 80% of the cells were then taking up DNA. And this was quite clearly present, not just on the surface, but inside the cells themselves. We then did the same thing with bone marrow derived dendritic cells, using a protocol that had been developed by other labs, showing that if you add FLT3 ligand and GM-CSF, you can actually induce CD103 positive BMDCs, whereas normal protocols just induced type 2 DCs. Importantly, these cells express very high levels of TIM3, so we're able to use this to evaluate the role of TIM3 in vitro in these cells. And what we found was that, consistent with our splenic DCs, when we added heat shock alone, we saw no DNA uptake by these cells. But when we added the TIM3 blockade in the presence of this essentially tumor cell debris containing DNA, we now were very readily able to detect DNA uptake by these cells. And this DNA was present within the cytoplasm as marked by GAPDH. This was not just limited to mouse cells in vitro. We wanted to see whether we could see the same biology in human cells. So for that case, we took human peripheral blood cells from healthy donors, and we did a dendritic cell isolation. Not shown here, but these cells express very high levels of TIM3, both in type 1 and type 2 DCs, allowing us to use these cells as we would splenic cells from the mouse. And we saw a very similar biology, whereas heat shock alone, in this case, heat shock from a human breast cancer cell line, we saw moderate levels of uptake by these cells. When we added heat shock in combination with this commercially available blocking antibody, we saw massive uptake by these human peripheral blood cells. We also did the same experiment using a collaboration with Tesaro before they were bought out by GSK. Their clinical antibody really had the same effect in terms of inducing DNA uptake by the cells. And when we looked at gene expression by the DCs, we could see that TIM3 blockade in the presence of this heat shock also led to upregulation of the same genes, CXCL9, 10, and 11, by these cells, suggesting we're seeing very parallel biology in human and mouse. So how is this process occurring at a molecular level? So I've talked before about some of the key ligands in terms of CCAM1, galectin-9, and phosphatidylserine. None of these really made sense in terms of how these ligand interactions would regulate DNA uptake. However, back in 2012, a Japanese group had also shown that TIM3 can bind to H&GB1. This was largely shown through in vitro assays and a few in vivo models. And I think other labs have trouble repeating this data, so there's some questions of how strong this interaction is. But this group at least showed that H&GB1 binds to TIM3 with roughly the same affinity as it does to RAGE, which is the other major H&GB1 receptor on the surface. And importantly, if you look at H&GB1, this is one of the most highly expressed molecules in the nucleus of cells. This is just showing staining in green here for H&GB1 in PYMT cells. So there's lots of it. It's in the nucleus. It binds to DNA, suggesting this was really the regulator of this DNA uptake and suppressive pathway we were seeing. So to evaluate this, we took advantage of a neutralizing antibody against H&GB1, again, commercially available. We added this to our experimental system. And then what we found right clearly is that heat shock in TIM3 lights up expression, in this case, allows for DNA uptake to occur. If we add this blocking antibody, we no longer see uptake by these cells. And then alongside the DNA uptake, we also saw in vitro that if we add this antibody, we no longer see CXCL9 expression. And then extending this out, we actually could take this heat shock, inject it into tumors, and track DNA uptake by the resident populations of macrophages and DCs, for example. And what we found was when we added heat shock into tumors from mice that had been injected with anti-TIM3, we were able to track uptake by these cells quite readily. And if we added an H&GB1 blocking antibody at the same time we injected the heat shock, we found that this uptake was dramatically reduced, although not completely eliminated. Most importantly, we found that H&GB1 was not only required for DNA necessary for DNA uptake to occur. We could actually add H&GB1 to purify DNA, and we now saw uptake occurring. So one of the caveats to the work, previous work in the area of sting activation in DCs, was if you take naked purified DNA and add it to dendritic cells, they don't take it up, they don't activate cGAS and sting. So it's never clear how they would then activate cGAS and sting pathways in vivo. And what we think is happening is that because if you purify out DNA, it's no longer bound to protein, it's no longer recognized by the cells, and you no longer get this uptake from occurring. So we acquired just simple commercially available beta DNA, which is only 500 base pairs long. It's labeled with rhodamine. We added it to the cells, and then we either did it alone or in the presence of this H&GB1 recombinant antibody, in this case commercial, but we also made our own as well. And what we can see here is that beta DNA in TIM3 does nothing. If you add beta DNA and H&GB1, you see some uptake by the cells. But really, if you now block TIM3, adding this mix of beta DNA and H&GB1, you now see uptake occurring. And this is just shown here in some representative images that we generated after the paper was published. You really see DNA uptake quite readily by the cells using the system. At the same time, while adding H&GB1 allows cells to take up DNA, it also allows the cells to then bind H&GB1 bound DNA to TIM3. So you activate both the uptake mechanism and the mechanism that blocks that uptake from occurring. And we did that using just a simple in vitro bead assay where we coated TIM3 protein onto the surface of beads and then measured binding to either beta DNA or beta DNA coded to H&GB1. And you can see here, while DNA alone does not really bind to the beads, we get quick increase in DNA binding once you add the recombinant H&GB1. So what we think is happening is that in tumors where the cells are either dying spontaneously or through irradiation, or in our case, through paclitaxel chemotherapy, this releases a level of H&GB1 and DNA bound in complex, as well as a host of other proteins that we found on the surface or bound to the DNA. When dendritic cells express TIM3, they're able to then bind to this complex, and that prevents uptake from occurring. If we then block TIM3, we are then, through some mechanism we are currently exploring, able to get DNA uptake to occur. This activates cGAS to produce this cGAMP, activates STING, leading to expression of type 1 interferons, which then signal back into the cells to activate expression of CXCL9. And as I showed in the first part of the talk, this allows then dendritic cells to increase their exposure of IL-12 to T cells and initiate a more effective immune response in these tumors. And we know this process here, at least in preliminary data, we know that this process is an endocytic one. So if we treat the dendritic cells with an inhibitor of dynamin, shown here in dinosaur, we can inhibit endocytosis by these cells. We completely block uptake, and this is just shown here for the control, showing that its endocytosis is blocked with this inhibitor in terms of transferrin. Whereas if we blocked other pathways, pinocytosis or phagocytosis, we didn't see any inhibition of this uptake. And then finally, what I didn't show here today, but we also showed in the paper, was that galectin-9, which is a ligand for TIM3, is also critically involved in this process. What we found is that if we add a Gal-9 antibody, and just shown here, we can actually ablate or reduce the clustering of TIM3 on the surface, resulting in reduced signaling, as you see by confocal. And what we think is happening is that by adding this Gal-9 antibody, we reduce clustering. We essentially reduce the avidity of this interaction, knowing that TIM3 binds to HTGB1 very weakly, but that these DNA and HTGB1 complexes are quite large. We may have very high avidity interactions, very low affinity, and by interfering with this clustering, we can then get the same effect we were seeing with TIM3. So if we add anti-Gal-9 antibody, these mice respond better to chemotherapy, dendritic cells take up DNA and they activate CXCL9, just as if we were adding a TIM3 blocking antibody. So if you were doing a Gal-9 blocking antibody as a therapy, we'd expect it to produce very similar results to TIM3 blockade in this one particular context. And so with that, I'll just leave up some kind of open questions that we have going forward. So we're very curious what role TIM3 has in terms of this expression by DCs and what role that has in other diseases. In particular, why do dendritic cells have a inhibitory receptor? It's constitutively expressed to block DNA uptake, but why do they have then a pathway for DNA uptake to occur? Why are they both allowing this process to occur and then blocking at the same time? What's the regulatory mechanisms and what's the reason for this? Particularly for us focusing on autoimmunity and trying to dissect that out, a role in those disease models. One of our primary focuses now is really trying to figure out how does DNA enter cells. We know this is HTGB1 dependent. We know we have an endocytic process. Does that mean we have an HTGB1 receptor? Or does it mean we have entire other pathway that we haven't identified? And just by binding HTGB1, we're able to interfere with that process through TIM3. So roughly a month or two after our paper came out in Immunity, the Kutcher lab also showed that if you ablate TIM3 expression by tumor DCs, that produces enhanced anti-tumor immunity. A phenotype that they did not observe when they ablated TIM3 in T cells. So in this case, in a tumor model where you have high TIM3 expressed by T cells, you still see the primary role of TIM3 was in DCs. However, in this case, they found that the absence of TIM3 really led to inflammasome activation. But given the potential for crosstalk between STING and inflammasome, particularly as it relates to AIM2 recognition of double-stranded DNA, there's lots of maybe potential interesting biology here to understand how these cells are responding and what the role of TIM3 is beyond just CGAS and STING. We're also hoping that we can understand, now that we know how TIM3 functions, can we then design therapies that take advantage of how TIM3 blocks DNA uptake, for example, by inducing therapies that cause a lot of HTGB1 or DNA to be released by cells. And finally, more long-term, we're really focused on understanding how, if there are other suppressive pathways in the tumor, obviously we know, for example, interleukin-10 is a primary suppressor of IL-12. We know that lipid uptake by DCs can regulate their dysfunction. There's sure to be certain metabolic pathways in tumor DCs that cause lack of maturation. And all of these are potential interests for the lab as kind of a big long-term project. So with that, I'd like to thank those that did the work. So here's the lab that we got together just after pre-Omicron, but post third dose. So we could actually go maskless for a couple weeks in that glorious time to celebrate a couple of publications and a thesis by our student. So everyone shown here was involved in the Immunity paper and as well as work on the cancer cell paper. primarily highlighting the work by Alvaro, who was the first author on both our cancer cell and immunity paper, but really it was a group effort, especially during the middle of COVID, doing revisions, where everyone pitched in to generate a figure or two for the paper. And of course, thanking our funding from the NIH, the Florida Department of Health, the DoD, and then early stage funding from Tesaro to work with some of the basic mechanistic stuff of TIM3. And with that, I will take any questions.
Well, thank you so much for an absolutely beautiful talk. That was really a phenomenal body of work, breaking new ground. So I'd like to start with the first question, which is, do you think that TIM3 could be an inhibitory co-receptor for TLRs? So TLR4 has been shown to be an HMGB1 receptor, and that can attribute to some functions of TLR4 in tumors. So have you looked at whether or not there's the close association or co-signaling between TLRs and TIM3?
Yeah, we haven't looked yet at signaling. We are trying to see whether with TLR4 may be involved in the DNA uptake process. So we have TLR4 knockout mice, and we're doing those now. But preliminary, at least, it doesn't seem to have a role. But whether it comes as a cofactor, that would be the confusing part. We may need to knock out 2 or 3 genes to completely ablate DNA uptake from occurring. So early data says no, but I'm not convinced that it doesn't have a critical role yet in the pathway.
That's very, very exciting work. And so I just had one more question, which is, you see that TIM3 is highly expressed in these tumor DCs. So are there circumstances where you don't see TIM3 expressed? I might have missed that.
No, for now it's consistently expressed by dendritic cells that are at least at the maturation stage of splenic DCs, lymph node DCs, human peripheral blood DCs, high levels of expression. I don't think we haven't looked at pre-DCs, so I'm not sure what stage they transition to having TIM3 expressed. And we know, I think that if we do classic FLT3 BMDCs, we don't see it expressed at all. So we think there's some role for GM-CSF there at driving expression.
So just to follow up to Judy's question, when you look at the tumor DC, do you see any heterogeneity between different population of dendritic cells there in terms of TIM3 expression?
Yeah, so it's definitely higher on the type 1 DCs. And that's consistent with, even in the spleen, you see high expression by both, but it's still 2 or 3-fold higher by the type 1 DCs. And that we see a pattern that's quite consistent across the board. So if you look at lung, lymph node, that pattern holds up.
So do this other DC contribute then to type 1 interferon production, since they have lower TIM3, like CDC2 or DC3? Do you know anything about it?
It's possible. I mean, we know if we eliminate it from the type 1 DCs, we lose our phenotype, but that doesn't say that there's also not an important secondary role in these other immune cells. We think that macrophages are probably not being activated, in that they would probably rapidly digest their DNA to prevent activation of cGAS and STING. But I don't think we know whether type 2 DCs are also being activated. At least in splenic DCs, when we do gene expression, we do see some upregulation of CXCL9 and 10, but we don't see a consistent upregulation of the type 1 interferons in those cells for a reason we don't quite understand. We know they take up DNA, but they're not inducing that cGAS STING signaling to induce the type 1 interferon expression, at least not at a high level.
Yeah, I think it's very fascinating. It could be partially, that some of the trials so far did not pin down any clinical effect. So it could be some of the mechanisms, right? Complication of this.
Yeah, I think there's a really nice preprint by a lab showing that they're developing like virus-like particles where they add the STING agonist there, and they show that those go into DCs and they induce anti-tumor immunity. and that helps avoid the off-target effects of sting on tumor cells from the work by the backbone group, or by potentially killing T cells directly through their sting.
Oh yeah, yeah. Well, I mean, actually, TIM3 targeting.
Oh,
not
TIM3.
this. TIM3, yes, but it's a
yeah,
it's
we're still waiting to
complicated.
see what those results look like from the solid tumors. I know Novartis has moved primarily to AML. But
Insight, unfortunately, wasn't successful. So
the Insight one,
anyway,
okay.
well, it was reported by FDA. Okay.
We have some questions in the chat. Jennifer, do you want to start?
Yeah, I was just wondering if anyone wants to turn their camera on to ask the questions. Hi, Dr. Pollard, any question or?
Yeah, I probably missed this. I just wondered about the trafficking of these DCs. It seemed that it really wasn't in the primary tumor, more in the spleen and perhaps lymph node. And I wondered if you've been able to track the DCs from the primary tumors. And I think these are all transplanted tumors and not spontaneously arising ones. That was another question I had. I also had a comment about one of the comments in the chat where somebody asked if there's LPS in the system. Well, there are reports of bacteria actually in the polyanimal model. I don't know if that's real. We've never really looked ourselves. There have been reports that bacterial colonization occurs.
So let's see. Second question first. So we did some of the early work in the transgenic model in terms of efficacy, but all the biology mechanistic work that was done in transplantable models. We did do trafficking measurements using the ZIA screen uptake to mark kind of tumor DCs, although we may miss some of those as they're going through and if they process the antigen quick enough. In that case, we didn't see any. We saw an actual like a 50% reduction in the movement of the ZIA screen positive CD103 DCs into the draining lymph nodes. Whether that's biologically relevant or not, we don't know, although it was relatively consistent across the experiments.
So do you think these activation is occurring by sort of exosomes or just sort of DNA aggregates which have been released and then accumulating in the lymph node or even in the spleen?
So yeah, we were, I forgot the name of the lab, but there's some nice work showing that the loss of DNA 13L or something causes autoimmunity. And that's through an exosome-based mechanism. And so that's those PIs suggested us to evaluate that. So we actually did the centrifugation steps, and then we quite rapidly lose our signaling through the heat shock when we get rid of the second spin. So by the time we get anywhere near the exosome stage, it's already lost. And so we can actually find it directly into the kind of organelle chromosome size centrifugation step.
Bits of DNA, bits of chromatin and things. Thank you. Very nice work.
Thanks.
Really enjoyed it.
Would anybody else like to ask their question directly, or we can read the chat? Okay. Well, I'll read the question from Tatiana Novobrantseva. She says, can you please discuss if you can target TIM3 specifically on DCs or CDCs? Also, can you discuss which epitope on TIM3 you expect to have the main anti-tumor effect? And is that on other cells? That's an interesting question.
Yeah, good questions. So I don't think we can specifically target them. Certainly every antibody developed so far targets all the cells. I would assume that there is some specificity in terms of the carbohydrate structure. That may give some specificity on a cell type basis, but I haven't seen any data on that yet. And certainly from all the available literature, galectin-9 binds to TIM3 on T cells and dendritic cells as well. But that is one possible area where they could, you could develop a therapy that targeted one particular aspect of cells. In terms of the epitope, great question. So this really work done by Anna Anderson's group has really defined how where the different antibodies bind and how they may function at a molecular level to block TIM3 function. Most of the blocking antibodies are designed to hit that phosphatidylserine binding site, which is shared by phosphatidylserine, HNGB1 and CCAM. And the only other major binding site is really, again, the galectin-9 sites there. So antibodies that either target the site directly or that bind near to and may change the conformation of that binding site, those seem to work as functional blocking antibodies. And there's also some clinical antibodies that seem to block galectin-9 and don't actually block binding of that site. And those seem to also work, at least in their in vitro systems, to activate T cells.
Interesting.
There's more questions in the chat. This one you sort of touched on, but Kelly Kirsten asks if besides the effect of anti-TIM3 on the CDCs, does it affect CD8 T cells directly? And do exhausted dysfunctional T cells in the PYMT model express TIM3?
So we see that dual TIM3 positive, PD1 positive population in about 3 to 5% of the cells. So they are there. We can't completely rule out that we're having some secondary effect on those cells through the TIM3 blockade. That's really going to require us to do the flux alleles, knockout mice. We're in the process of that. But the Kritcher lab just showed that if you knock out TIM3 and DCs, you get spontaneous anti-tumor immunity. And if you did it on CD4s or CD8s, you didn't see that same effect, suggesting it really is a DC specific mechanism. That said, the Lawrence King group at Pitt also published just recently in Cell Reports, where they did Treg specific knockouts of TIM3. And there they found that if you knock out TIM3 from Tregs, you had those tumors grow less well in the mice. So I don't think we can say that it's entirely dendritic cell based. Definitely there is some, I think there is going to be some T cell role here. And importantly, there are humans with TIM3 mutations, and they don't develop, at least in the few case reports that have been, they don't develop autoimmunity. They actually develop T cell based cancers. So advanced proliferative disease, suggesting it does have a really functional role in T cell biology, as opposed to, at least in these contexts, in dendritic cells.
I have a question that you just sparked. So you showed TIM3 is highly expressed on macrophages in the tumors. So have you got any idea about the role in macrophages in your system?
We have not seen any change in their biology as of yet. But I think the second or third paper from the Kritcher group by Anna Anderson, when she was there, was looking at microglia, for example, and they showed that TIM3 regulates the activation of these cells. So this is back Nature Immunology back in the early 2000s. So there is that, and there are miscellaneous papers after that suggesting that TIM3 has a role in myeloid activation, but nothing I think that's been quite cleanly shown, and what it's doing in those cells is not clear at this time.
Well, you did kind of address this one question from ACL, but I think maybe it could be more detailed. So that this person was interested in the internalization of TIM3 that you by endosomes. So do you have any more detail of the type of endosome?
I would love to know that information. My poor postdoc has been trying to stain for these different early and late stage lysosomes, endosomes for some time. It's a little hard because the cells are so small. So there's not much cytoplasmic space to analyze. And what we can see is that it's a really fast process, which surprised us. And then we actually look at the literature and they're like, oh yeah, it takes like 30 seconds to a minute to happen. So this is an extremely fast process that it is really hard to track. We can see uptake within minutes of these cells being exposed to high levels of DNA. And we haven't really seen much co-staining in lysosomes and endosomes. So what we think is they may be escaping quite rapidly from the very early endosomal stage. So maybe, and this is conjecture on my part, but the work from the Catena de Souza lab has shown that CLEC9A allows very rapid escape from the endosomes to avoid lysosomal digestion. And so perhaps TIM3 or some other complex there has a similar role. HNGB1 has also been shown to mediate escape from endosomes, and that may be involved in the release and into the cytoplasm as well.
Mhm. So you're basically saying that if you have a knockout TIM3 or blocked TIM3, you have slower or no faster release from the endosome. Is that what you meant?
Well, I don't think it's TIM3. I think TIM3 is blocking the uptake altogether.
Yeah.
So it's blocking binding.
It's not that TIM3 dependent, it's what happens after. But when you say fast release, what are you comparing it to? That's just what I want.
More that not that I'm comparing to any particular uptake system, but knowing that it happens within a couple of minutes at least. We see cytoplasmic localization within a few minutes.
Okay.
And so we have trouble actually finding that, assuming it's endosomal, we have trouble actually seeing that co-localization in the endosome.
Yeah.
Brian, I have a question about HMGB1. So if my understanding is that it's different, has different post-translational modifications depending on the source of the cell it comes from. So whether it's secreted by immune cells or from necrotic cells, apoptotic cells, do you see any differences in responses based on the source of HMGB1 and not related to the question related that asked about the bacterial LPS?
Yeah, so HMGB1 can be, it has a disulfide bond, so that regulates its redox state. It can be acetylated. I think it can be methylated or something else too. So lots of modifications that it can occur. Certainly the redox state is of interest to us, but we haven't dived down into that in greater detail. We have generated the HMGB1 mutants that have the inability to form that disulfide bond, but we haven't seen that alters binding to binding to TIM3 as of yet. It may binding to DNA, so that may be where the regulation comes in. So it's been shown that the different redox states have different binding properties to DNA, and that's mediated through the conformation of the little acidic tail on HMGB1 that then kind of folds up the protein depending on the redox state. So we think there's some interesting biology there, in particular how HMGB1 gets out of the cell with DNA, if this is an active process or are we just dealing with lysis of the cells. But we're really in early stages of that in the lab.
There's a couple more questions in chat, and then I think we can call it a day. Sorry, I can't pronounce the name. One question then is, how about the different effect of anti-TIM3 on the human tumor cell in immune deficient mice? So I was thinking that too. Is there a way you can analyze some things that are going on by looking at the effects in immune compromised mice? So
I mean, we've done, if we do like CD8 depletion, so those are immune compromised, and then we don't see any efficacy in that situation.
yeah.
So I'm not sure about the reason to do it. I can see doing it in the humanized mouse model where you really want to ask, are we having effects, biological effects on human T cells and human DCs? Although I think that's the complexities of that make me prone to just hope they finish the clinical trial, so I don't have to do any of those extremely expensive studies, which are always hard to analyze anyway because the system's so complex.
So I mean, that leads me to think of something. Is there a way you could deplete macrophages to see whether, specifically without affecting DCs, to see whether there's any like dual role for macrophages and DCs, like a 1-2 punch kind of thing?
Yeah, I'm trying to remember if we did any CSF1 neutralization with TIM3. I don't think we have, so I don't think we know yet whether there's a role for macrophages there. Although if we depleted them, we may see some of the same, we know that also improved response to paclitaxel in our system. So we may just have the exact same effect regardless of how we get there.
Yeah.
But yeah, I definitely have interest, and there's some of my students are interested in what macrophages are doing in terms of TIM3 and why is it expressed at high levels, and does that matter? It's hard to imagine it doesn't do something, but at this point we don't have any data to suggest it has a role. But now that we have the TIM3 flux mice now, so we can actually knock it out of macrophages, knock it out of DCs, and see if there's some biology there.
Yeah, nice.
Very cool. I can ask the last question in the chat. This is related to the effect of anti-TIM3 on the activation of T cells through the migratory CDC1s in the tumor draining lymph nodes. So for example, the CXCL9 production also favored the TCDC interaction in the draining lymph nodes.
Yeah, that's a fantastic question. So there's some really nice work, and I'm blanking on the name, showing that DC expression of CXCL9 in the draining lymph nodes mediates these T cell interactions from almost a decade ago. And it was actually that kind of idea in that paper that inspired us to look at local interactions mediating the same biology. So that was one of our first thoughts as well. So we tried doing FT720 to block lymph node egress in our system. And I didn't show the data here, but that doesn't change the response to therapy at all. We also did CCR7 deficient bone marrow chimeras. So then we can also, we allow immunity to develop, and then we deplete out the CCR7 positive DCs. In that case, we also could see response to TIM3, but at the later stages of therapy, it was slightly reduced. So we think there may be a role for DC migration, maybe not in response to TIM3, but just as a basal level being required for therapy to be efficacious, whether it's TIM3 blockade or it's PD-1, similar to the JAXA paper that just came out in Cancer Cell like a few months ago.
Thank you so much for an absolutely wonderful presentation and very stimulating work that explains a lot of biology too. So I wish you continued great luck on your work. It's just phenomenal, and hope to see everybody next month and then again for our retreat. So,
yeah, thanks so much, Brian. Michael Karen is next month.
Great.
Thank you for the invitation, Jen, Judy, Lisa, Dimitri, and yeah, looking forward to next month's talk as well.
Okay. Right.
Thanks, Brian. Good to see you.